In this video, we'll discuss about phloem, which is the principal food conducting tissue in the plant. So, what phloem do is that they transport the metabolites which are synthesized in the leaves during photosynthesis to other parts of the plant such as roots, flowers and fruits. Okay, so this phloem can be differentiated or classified into two based on the origin. Primary phloem, which are derived from procambium, that is the meristematic tissue, and secondary phloem, which originate from vascular cambium, the cambial ring which is formed during the secondary growth of a plant. Primary phloem can again be classified into two, protophloem and metaphloem. Protophloem is the first formed phloem. Primary phloem can again be categorized into two, protophloem and metaphloem. Protophloem is the first formed phloem tissues. They are non-functional, and metaphloem, which is the later form, is the only conducting phloem tissue in an adult plant. Then based on the position, the phloem can be categorized into intraciliary phloem and interciliary phloem. Intraciliary phloem, it consists of internal phloem and external phloem. Internal phloem means the phloem which occupies a position internal to xylem. Means if we take the section of a plant part, and if the xylem is situated towards the periphery and phloem is situated towards the pith, such types of phloem can be said as categorized as internal phloem. And external phloem means the phloem which occupies a position external to xylem. Means if the xylem tissue is situated towards the pith and phloem is situated towards the periphery, that is the epidermis, then that type of phloem can be categorized as external phloem. So both this internal and external phloem, they constitute the intraciliary phloem. In the picture, look into the vascular tissues, vascular bundle, and you can see it is labeled as IPH and EPH. IPH is the phloem tissue which is seen towards the towards the pith, that is to the internal to xylem, whereas EPH is the phloem which is the external phloem which occupies a position external to xylem towards the cortex and epidermis. Then coming to intraciliary phloem, the phloem which can be seen as embedded in the xylem tissue. It normally occurs in secondary vascular tissues. So the phloem tissues can it can be seen as patches as shown in the second image B, or the xylem and phloem is arranged as alternating bands. Okay, so in both the scenario, the phloem is seen inside the xylem tissues. So such type of phloem tissues are known as interciliary phloem or included phloem. Phloem tissue is composed of four different kinds of cells: sieve element, companion cells, phloem parenchyma and phloem fibers. So first component is sieve element. Sieve element is of two types, the sieve cells and sieve tube members. Sieve cells are seen in plant groups except the angiosperm, means gymnosperm, ferns and hostels. In all these groups, the phloem consists of sieve cells. Okay, but in angiosperm, it is sieve tube member, also known as sieve tube element. So the sieve element is of two types. And this sieve element, either the sieve cell or the sieve tube member, this is the chief food conducting cell in the phloem tissue. And it is elongated cells, and the wall has a group of pores which is known as sieve area. Okay, so the sieve, it resembles the sieve, the utensils which we use in the kitchen. So the name sieve cell and sieve tube member. Okay, so the wall consists of pores in it, which is similar to that of the perforation plate, more or less similar. The structure is entirely different, but when we look into the morphology is similar, almost similar. Okay. Then coming to sieve tube member, it is a long tubular cell. So here is a picture of sieve tube members. It is an elongated cell, tubular cell, and they are living cells. It is living, but it is non-nucleated. There is no nucleus and other organelles, cell organelles, which is essential to maintain the cell in living condition. Okay, so it is devoid of, the sieve tube member is devoid of cell organelles, but it is living. It consists of a thin peripheral cytoplasm and a large vacuole. And it is characterized by the presence of sieve plate. Sieve plate is the part of wall with sieve area, means that part of the wall have large pores in it. Such wall part is known as sieve plates. And the sieve plates are of two types, simple sieve plate and compound sieve plate. Simple sieve plate means the sieve plates having only a single sieve area. And compound sieve plate is the sieve plate having two or more sieve area. Okay, so in the picture, the first one, the picture to the left, it is a diagrammatic presentation of compound sieve plate where there are 4 sieve areas arranged in each end. Whereas simple sieve plate, it consists of only a single sieve area at each end. So the diagram to the right is that of simple sieve plate. And the sieve plate usually occurs on the end walls, and each sieve tube members are arranged end to end, and together they form a sieve tube. And inside the sieve tube element or sieve tube member, there is a protein which is known as P protein, phloem protein, and slime. So together they form slime plugs which accumulates in the sieve area if a sieve tube member get damaged or they are infected, so that this infected or damaged sieve tube member will be isolated from other members by the accumulation of slime plug at the sieve plate. And we said the sieve tube member, it is living, but it is devoid of cell organelles. So to keep that element in living condition, it is associated with a specialized parenchyma cell which is known as companion cell. And the sieve tube elements together with companion cell is known as sieve tube companion cell complex or sieve element companion cell complex. So the second component is companion cell, the second component of a phloem tissue. And this companion cell is a specialized parenchyma cell, so that it have all the components of normal parenchyma cells including nucleus. So all the activities of the sieve tube member is controlled by the nucleus in the companion cells. And the companion cells communicate with the sieve tube member through plasmodesmata. And the plasmodesmata is the cytoplasmic connection between the cells. So the all the elements which are necessary to maintain the sieve tube member is transported from the companion cell to the sieve tube member through the plasmodesmata. And actually the companion cell and the sieve tube members, they are derived from the same mother cell. So a mother cell which undergo unequal division give rise to a larger daughter cell which becomes the sieve tube member, and a smaller one which becomes the companion cell. So here is the diagram of the development of sieve tube element and the companion cell from a single mother cell. And next is sieve cell. Sieve cell, it is the sieve element which is seen in gymnosperms, ferns and other plant groups except that of angiosperms. So it is also elongated cells and have tapering end walls, and they lack P proteins, companion cell and sieve plates. Okay, so instead of companion cell, the sieve cells are associated functionally with that of albuminous cell, also known as Strasburger cell. And the sieve cell and albuminous cells, they are not necessarily derived from the same mother cell. Unlike that of the sieve tube member and companion cell, they are derived from a single mother cell, but that is not the case in sieve cell and albuminous cell. Okay. And the third component in phloem is phloem parenchyma cells. And in primary phloem parenchyma cell, the main function is storage. It stores various substances such as starch, tannin, and crystals. Whereas secondary phloem parenchyma cell are of two types: axial parenchyma cell and ray parenchyma cell. Axial parenchyma cell is concerned with the storage, whereas ray parenchyma cells are for the lateral conduction. And the last component, phloem fiber, we have discussed the phloem fibers in the sclerenchyma fiber in the simple tissue. So you can refer that video on simple tissue for the details. And the terminology associated with the complex tissue is transfer cells. So transfer cells are specialized parenchyma cells, but the parenchyma cell have certain inner cell wall ingrowths which is similar to the cristae in mitochondria. So these ingrowths are deposited on primary cell wall. So the ingrowths are considered as special secondary wall. And the main role of transfer cell is short distance transport of sugar or solutes. So this is about phloem. Various questions regarding the phloem and xylem are usually asked in exams. So you can refer the short videos for the previous year's questions from old question paper. And if you have any doubt, please do ask in the comment section. Thank you for watching. Please do subscribe. Thank you.